Hello everyone and welcome to the fourth session of week 1 of our course on pharmacognosy and phytochemistry. In this session, we will dwell little deeper into set of compounds called as terpenes. We will understand them, and based on our previous session, that is our biosynthetic building blocks, we will also try to synthesize a few terpenes from this biosynthetic pathways. So let's learn what are the terpenes compounds and how they are biosynthesized. So if you see the building block that we discussed previously, that is the C5 building block, that is made up of isoprene. So isoprene building blocks have a rule that whenever you have one isoprene unit, it carries a formula of C5H8. So whenever there are multiples of this, or as isoprenes go on adding, you just have to multiply. So if my single building block is C5H8, when there are two building blocks, it will become C10H16, that is C5 into twice C10 and H8 into 2, that is H16. Similarly, depending upon that, in nature you have various terpene compounds. So we will study C5 building block in detail. So C5 building block is made up of isoprene unit, and this isoprene unit can be brought in by two different biosynthetic precursors. One is mevalonic acid, you can see here, which comes from the acetate mevalonate pathway, and the second, what is your deoxyxylose phosphate, which comes from methyl erythritol phosphate pathway. Now we are going to learn both of these pathways to derive terpenes. Now what are terpenes? Terpenes are C5 derivatives, and based on how many C5 compounds are present, they get their nomenclature. To start with, just to take the first example, hemiterpenes, hemi means half. So when I say C5, that is the first unit or the basic building blocks, they are referred to as hemiterpene. So if C5 is half, the full one is called as monoterpene, and it will be twice. So C10 derivatives are called as monoterpenes. Now adding 5 to each of them, you will get C15, which is the next building block, and they are called as sesquiterpenes. So you have your hemiterpenes, which is C5, you have your monoterpenes, which is C10, then you have C15, which is your sesquiterpenes. Now surprisingly, in nature one more phenomenon happens, that is there are compounds especially which are aromatic in nature, which are coming from isoprene unit, they don't fit in multiples of 5. They are called as homoterpenes, and generally they are C5 multiples plus 1 carbon. So you might come across compound which is like C11, which is C10 plus 1, or you might come across compounds such as C16, which is a C15 plus 1. They are called as homoterpene derivatives. What happens is they are terpenes derivatives. They are generally derived as multiples of 5, but undergo some elimination and some type of reactions to become just a carbon extra to the terpenoidal derivatives. So they, being still terpenoidal in their biosynthesis, are referred to as homoterpenes. Now going ahead, if mono is C10, then you duplicate it, then you will get is twice monoterpene, which is your diterpene, that is C20 derivative. In addition to that, just add 5 to it, you have what is called as sester terpenes, and then just add one more 5 to it, and you will have what are called as C30 derivatives. Now C30 derivatives are referred to as triterpenes. C10 is mono, C20 is di, and C30 is your triterpenes. Now if you go to add 10 more to it, you get C40 compounds, which are your tetraterpenes, and tetraterpenes are very big. In the previous session, we saw an example of carotene. Now this carotene compounds such as lycopene are definitely your C40 compounds, which is made up of about 8 such isoprene units. So 8 5s are 40 makes tetraterpene compounds. So all of these terpenes come in from these two basic metabolic pathways. So let's understand them. Now moving to acetate mevalonate pathway, as the name indicate, we get this pathway from acetyl-CoA, and this acetyl-CoA comes from what is called as Embden-Meyerhof pathway. This is the same pathway which converts your glucose into pyruvate, and thereafter your pyruvate is converted into acetyl-CoA. Now this acetyl-CoA pathway or acetate mevalonate pathway or isoprenoid pathway or HMG-CoA reductase pathway are synonymously used and essentially means the same, that is your acetate mevalonate pathway, which involves intermediates such as mevalonic acid. Now this pathway is used for biosynthesizing compounds such as terpenes and also steroids, especially your hormonal compounds. It is found in cytoplasm and starts with a simple molecule of acetyl-CoA. This acetyl-CoA, 3 such molecules of which add together to form a mevalonic acid, and thereafter you get your isoprenoid units, which are your isopentyl pyrophosphate and dimethyl allyl pyrophosphate. So what happens chemically? Let us try to understand it. So we start with a simple molecule, that is your acetyl-CoA. Okay. Now when you see acetyl-CoA, this acetyl-CoA is a acetate which has been added to an enzyme. Okay. Now what happens here is this acetyl-CoA which gets added to an enzyme gets condensed with another molecule of acetyl-CoA in presence of an enzyme, that is acetyl-CoA thiolase. Thiolase kind of cleaves the SCOA group and your acetyl-CoA joins to each other. So you find this functionality joins here to the methyl, this is the methyl of next acetyl-CoA, and you get what is called as acetyl-CoA. Now this acetyl-CoA undergoes what is called as aldol addition, and one more molecule of acetyl-CoA is involved in the process. Now this is done in presence of an enzyme which is called as hydroxymethylglutaryl coenzyme A synthase. Now HMG-CoA synthase is a very important enzyme and it is involved in formation of molecule which is called as beta hydroxy beta methyl glutaryl CoA. Okay. So if you see, you have, this is the typically beta carbon. So you have your beta hydroxy beta methyl glutaryl CoA. Now you can clearly see what has happened is this particular oxygen that of the ketone group has got converted into a OH, and then you add this molecule. I have kept it as read out here, and you can see it carefully how it gets added. So this is the CH3, this is the C part. So it gets added, and what we have is now C2 plus C2 plus C2, that is we have 6 carbons. So this is my first acetyl-CoA, this is my second acetyl-CoA, and this is my third acetyl-CoA, which is kind of condensed together to form beta hydroxy beta methyl glutaryl CoA. Now this undergoes what is called as a reduction as well as elimination. The reduction happens here, and as a result you can see this getting converted into an alcohol. Okay. And then what happens is, you can see, because the reduction happens, this enzyme is called as a reductase. This happens in presence of a NADPH, a kind of cofactor. Now this produces one of a key intermediates in your isoprenyl pathway, and that is mevalonic acid. This is where your acetate mevalonate gets its name from. Now in some cases you might find it represented as a anion, that is mevalonate. That's also perfectly fine. I've put it here as mevalonic acid. Okay, so what happens here is, in your case of mevalonic acid, you might this mevalonic acid further gets phosphorylated, and phosphorylation often happens in presence of enzyme called as kinase. Now this kinase phosphorylates it at the 5th position. We initially saw at the 5th position the carbon got reduced. Now what happens is, this hydroxy further gets phosphorylated. Now this phosphorylation varies from organism to organism. In majority of the cases, you get what is called as 5-phosphomevalonic acid. So just to recollect, you have 3 molecules of acetyl-CoA condensing to form a 6 carbon derivative, which is your hydroxymethyl-glutaryl-CoA. Now this hydroxymethyl-glutaryl-CoA undergoes reduction to form mevalonic acid, and thereafter phosphorylation to forms phosphomevalonic acid. Now this phosphomevalonic acid, like I said, depending upon the organism, there are different postulates. One postulate says that it gets phosphorylated one more time at the 5th position to form diphosphomevalonic acid. One postulate also suggests that instead of at the 5th position, it might phosphorylate at 3 position, giving rise to 3,5 biphosphomevalonic acid. In some cases, it is suggested that the phosphorylation is eliminated first, the reduction happens, and then it gets rephosphorylated. But going by the most abundant rule, what we say is the phosphomevalonic acid gets phosphorylated one more time in presence of an enzyme 5-phosphomevalonate kinase, and what happens here is you get your diphosphomevalonic acid. Now this will undergo decarboxylation. So we are going to lose this. We initially had 6 carbons. Now a decarboxylation means a carbon less. So we are going to have effectively 5 carbons after decarboxylation, which we precisely want because our isoprene is a C5 unit. Not only that, when this decarboxylation happens, there is also elimination. The elimination of this hydroxy with the hydride from here leads to formation of double bonds. So this may undergo elimination and resulting in a double bond, and that's where we get our compound isopentyl pyrophosphate. Now this isopentyl pyrophosphate may undergo isomerization to form what is called as dimethyl allyl pyrophosphate. So these are our key intermediates, that is our isopentyl pyrophosphate and dimethyl allyl pyrophosphate, which are involved in synthesis of all our terpene derivatives. Going to the second pathway, that is your methyl erythritol phosphate pathway. Now this occurs in some bacteria, some eukaryotic parasites, as well as plastids in the plants, which produces chlorophyll. So your chlorophyll biosynthesis or most of your terpenes which are involved in your electron transport reactions mostly are prepared in plants by methyl erythritol pathway. Basically your methyl erythritol phosphate pathway. Now the intermediates which are required as a starting material for this are your pyruvate and glyceraldehyde 3-phosphate. Your pyruvate and glyceraldehyde 3-phosphate are also products of your Embden-Meyerhof pathway, which is nothing but your glucose pathway. So here in this particular pathway, your glucose gets converted into your pyruvic acid, and glyceraldehyde 3-phosphate is one of the intermediate during the process. Now both of these molecules condense to give you a key intermediate, which we call it as deoxyxylulose 5-phosphate, and this deoxyxylulose 5-phosphate then undergoes rearrangement to form a isoprenoidyl group. This isoprenoid then polymerizes to give compounds such as chlorophyll, carotenoids, tocopherol, prenylquinones, and so on. So let's understand this pathway. So a molecule of pyruvate condenses with glyceraldehyde 3-phosphate. Now what happens in this process is, you can see your pyruvate being condensing here, and our CO2 gets eliminated. So when CO2 gets eliminated, this joins here. Now you will see one more process being happening, and that is this aldehyde gets converted into an alcohol. So you will see pyruvate condenses with glyceraldehyde 3-phosphate, undergoes a decarboxylation in presence of an enzyme, that is 1-deoxyxylulose 5-phosphate synthase. That is, it is involved in the synthesis. Now once this synthase is involved, the second step what happens is it undergoes reduction as well as isomerization. So reduction of what? So if you see here, there is a ketone. This ketone gets converted into alcohol. Okay, so you have a reductase and you also have isomerase. Now what happens, or where is the isomerization occurring? You have a methyl here. This methyl shifts to this carbon. Okay, so you will see the methyl is shifted to this carbon, and the ketone which was there on this carbon has now got converted into alcohol, and that is done in the presence of enzyme, that is 1-deoxyxylulose 5-phosphate reductoisomerase, because reduction is also happening as well as isomerization. Now this gives rise to compound methyl D-erythritol 4-phosphate. Now this methyl erythritol 4-phosphate adds a cytidine triphosphate. Now in presence of cytidine triphosphate, you will see an adduct being formed, and this is the cytidine adduct. The compound name is 4-diphosphocylcytidyl 2C-methyl D-erythritol. Now this compound further undergoes one more phosphorylation in presence of an enzyme kinase, more specifically 4-diphosphocylcytidyl, again 2C-methyl D-erythritol 2-phosphate kinase. Now this kinase phosphorylates here, and as a result you have 3 phosphate groups in this. So the resultant compound which is formed is 4-diphosphocylcytidyl 2C-methyl D-erythritol 2-phosphate. Now this molecule carrying 3-phosphate undergoes a process of cyclization, but in order for that cyclization to happen, an elimination is must. So in the process we lose what is called as cytidyl monophosphate. So here your cytidyl monophosphate is lost or eliminated, and we are left with the compound with just 2 phosphate groups. Now these 2 phosphate groups cling to each other to cyclize. So you get what is called as a cyclodiphosphate adduct. So in the presence of enzyme 2C-methyl D-erythritol 2,4 cyclodiphosphate synthase, you get a cycloadduct, that is your 2C-methyl D-erythritol 2,4 cyclodiphosphate. Now what happens here is another interesting phenomenon, and that is kind of conversion with the removal of water molecule. So here you will see that carefully this is being eliminated along with this moiety. So it's a kind of water elimination simultaneously, and this gives rise to a double bond out here. So I have my CH2OH, I have my CH3 and a double bond, and now you can visualize your isoprene. So this happens in presence of an enzyme synthase, that is 1-hydroxy-2-methyl butynyl 4-diphosphate synthase. So as a result you get this compound which is 1-hydroxy-2-methyl butynyl diphosphate, which is very close to isoprene but just having 1 hydroxy extra. Now this hydroxy is removed by reduction, and the enzyme involved in this process is 1-hydroxy-2-methyl butynyl 4-diphosphate reductase. Once the reduction happens, you get the same isomers which came in from your acetate mevalonate pathway, that is your isopentyl diphosphate and dimethyl allyl pyrophosphate. So you get dimethyl allyl pyrophosphate, which kind of isomerizes and can be converted into isopentyl diphosphate. Now since the isomers are common, from here on we take the pathway as a common step. So we call it as a terpene biosynthesis. So 2 such building blocks, you can clearly see this. This is a dimethyl allyl pyrophosphate, and this is attached to your isopentyl pyrophosphate. In the process, it will undergo phosphate elimination, and since these two groups are being added up, this C5 plus C5 is C10, and C10 is your geranyl diphosphate. So, C5 plus C5 adds together in presence of an enzyme geranyl diphosphate synthase to give you geranyl diphosphate. Now, to this you can have one more isoprene unit being added. So, when one more isoprene unit is being added, again it will undergo phosphor elimination, and then you get what is called as a farnesyl diphosphate. Again it is easy to visualize. This is dimethyl allyl pyrophosphate, this is isopentyl pyrophosphate, this is isopentyl pyrophosphate. So, you have 3 C5 units. So, this is C10, this is C15. Now, here you can add one more to this, and what will happen? Your C15 plus 5, you will get your C20. So, again you have your dimethyl allyl pyrophosphate, isopentyl pyrophosphate, isopentyl pyrophosphate, isopentyl pyrophosphate to give you geranyl diphosphate. Now, in some cases, this monomers may dimerize to give you adducts. So, say for example, your geranyl diphosphate can be a dimer of C10, whereas your farnesyl, that is C15, may dimerize in the following manner. So, I just remove this phosphate and I take this carbon and I attach to this carbon. Now, generally what happens here is we saw previously what is called as head to tail condensation, that is we say 1, 2, 3 is head and 4, 5 is tail, which is attached to next head 1, 2, 3, 4, 5. Now, which is attached to next head, which is 1, 2, 3, 4, 5. But what happens here? If I am just talking about the head 1, 2, 3, 4, 5. Now, this is a 5, 5 attachment. So, this is a tail to tail condensation, and like we saw here the molecule shows a different kind of symmetry. So, in presence of enzyme squalene synthase, this molecule will join and fold. Now, if you see, the foldings are very proper, and then it will undergo formation of what is called as squalene epoxidase, and thereafter give you, I am not adding any carbon, I am just joining the dots, and you can clearly visualize that if I just join this double bonds, I break this double bonds and join this molecule or cyclize this molecule, I will get steroids. So, squalene is one of the key intermediate which is involved in steroidal synthesis. It is also involved in the synthesis of triterpenoidal compounds. So, from here we will get our steroids, we will get our triterpenoids. And what we require in the process is C5 into 6. So, you have C15 which is C3. So, we have C15 which is C5 into 3. So, 2 of such molecules. So, we call it as C5 into 6, which is your C30. Now, moving further, what might also happen is this big bulky molecules. You remember C20 plus C20. So, C20 may join to each other to form a big bulky C40 groups. So, this is done in presence of enzyme called as phytoene synthase. Now, this phytoene synthase, when it joins to each other, will just form a plain linkage. You need unsaturation. You will see naturally when I am talking about tetraterpenes, I am talking about carotene kind of compounds, and then when I am talking about carotene, these are colored compound. The color intensity depends on double bonds. So, in nature you will find most of the carotenes with double bonds. So, this synthase, it undergoes few more unsaturation in presence of enzyme phytoene desaturase to become what is called as carotene, which is there in your carrots. Now, this carotene will undergo still unsaturation to form lycopene, which is an intense red pigment which is present in your tomatoes. And then you will see cyclization. Now this cyclization happens in presence of enzyme called as lycopene cyclase, and it gives rise to both alpha and beta carotene. For convenience I have just put here beta carotene, but you can visualize it, it is very simple. You can just see it is the same thing. So I am just starting it from here, 1, 2, 3, 4, 5. I cut it here again, 1, 2, 3, 4, 5. I can cut it here 1, 2, 3, 4, 5, and I can go till here. So you can see the same kind of 4 isoprenes are there on each side, which are joined to each other here by a tail to tail linkage, is an important distinguishing characteristic of your tetraterpenoidal compounds. Now going to few examples of compounds. So when I am talking about hemiterpenes, hemiterpenes are C5H8 compounds. Now when I am saying terpenes, I mean hydrocarbons, but in some places you will see the word terpenoids. Terpenoids is when your terpenes are oxygenated. Carotenes is when it is hydrocarbon, carotenoids is when they are oxygenated. So whenever they are oxygenated, you replace the word terpenes by terpenoids. So example of hemiterpene is your prenol. In this case it is isoprene, but when it is prenol you call it as terpenoid. Now again one more example of isoprene is, you can easily see here, isovaleric acid, the same functionality. So this is my 5 here. Now we go to monoterpenes. Monoterpenes are C10. Now if you go to see monoterpenes as C10, there might be cyclized. So I am just giving you one example. So 1, 2, 3, I get here 4 and 5. Now if I cut it here, I can see the second one, 1, 2, 3, 4, 5. So this is basically 2 isoprenes joined in this manner. Similarly, you can do this for terpenol as well, it is very similar. You want to try the next one is your sesquiterpenes. Sesquiterpenes have 3 isoprenes. So by their nature they are C15 H24 compounds. An example of that is your humulene and farnesol. So you can see here, okay. Precisely 3 molecules of your IPP joined to each other to give you humulene. Now when you see monoterpenes or sesquiterpenes, they are low molecular weight, they are aromatic, they have a nice essence. So if I am talking about lemon or limonene, it gives you a strong smell of oranges. When I say terpenol, it is your turpentine smell. So this monoterpenes and sesquiterpenes as well as your hemiterpenes are volatile compounds by their nature, but as the molecular weight increases, their volatility decreases. So next you have diterpenes. Diterpenes are like your C20 compounds. So if you go to see something like your retinol, which is your vitamin A derivative, again we can visualize them. This is 1, 2, 3 for us. 4, 5, I cut it here, 2, 3 again, 4, 5, I cut it here. So you can see this is 1, this is 2, this is 3 and this is 4. So you have 5 into 4, which is your C20. Similarly, if you see your phytol, phytol is also a C20 derivative. Similarly, for your squalene, we just did it, we represented in a cyclized manner, but you can represent it in the linear manner. Here I have marked the isoprene, so you can see it clearly. Joined to each other by tail to tail condensation. Now you have your sesterpenes, example is your geranyl farnesol, that is C25 compounds. Then you have your triterpenes, squalene, we have done. Tetraterpenes, lycopene is an example if you recollect, which is there in your tomatoes, which is there in your watermelon. Then you have your beta carotene, which is there in your carrots. Then you have your polyterpenes. Your rubber latex is an example of a polyterpene, which is actually a polymer. Now, in addition to that, you also have a little different or additive compounds called as meroterpenes. Now, meroterpenes are terpenes in which the terpenes have been attached to slightly different molecules. They are not pure terpenes. So, they are terpenes plus something else. So, take for example bakuchiol, which is nowadays there in your cosmetic creams, which is replacing your retinol. So, you can see a typical head out here. Okay, that's the one, and then you can see the second one, 1, 2, 3, 4, 5. But you can see this molecule is not a terpene. So, it is attached to something else. Similarly, if you see this molecule which is there in your cannabis, that is your tetrahydrocannabinol. Okay, you can clearly see a terpenoidal group out here. We just represented it previously under the monoterpene. So, you can see 1, 2, 3 head, 4, 5. And then the second terpene 1, 2, 3, 4, 5. But from this side, it's no longer a monoterpene derivative or no longer has isoprene units. So, you can say it as an adduct. In addition to that, your polyterpenes occur in nature also as some of the monoterpenes, meroterpene derivatives. This include your ubiquinone. So, you can see in ubiquinone, which is involved in your electron transport chain, the terpenoidal 1, 2, 3, 4, 5 is there, and you will see that numerous such molecules, that is, it is a repeating monomer which is there, and this is attached to a benzoquinone nucleus. Whereas when you see tocopherols, or which are called as your vitamin E derivatives, which are your natural antioxidants, again in that you will see a good symmetry of isoprene units. So, now you can easily draw. So, 1, 2, 3, 4, 5. You will see that this unit is little no longer a terpene moiety. So, that tail or hydrophobic part of tocopherol is being contributed by your isoprene units. So, these are few examples of terpenoidal derivatives. They are biosynthesis. What we learnt is acetate mevalonate pathway and also your methyl erythritol phosphate pathway. Your methyl erythritol phosphate pathway is mostly used for production of plant pigments, whereas when you see your terpenes and aromatic compounds, it is generally the acetate mevalonate pathway which is used. So, here are a few references if you wish to learn more about this biosynthetic pathways, and thank you everyone for your patient listening.